Hello and welcome to SCP 105's latest discussion and lecture that you'll be watching as part of our ongoing virtual lecture series. This is the third week where we've been discussing various aspects of life in the universe. First week, we started off by introducing some concepts that are important for the class. In the second week, you saw a lecture where I discussed life throughout the universe and how we might discover it and where we might find it and what it might be made out of. And there we introduced a definition of life called the biochemical definition, a definition that relies on two chemicals to do the different processes of life. And those two chemicals are nucleic acid and amino acid. As you combine amino acids together, you end up creating proteins. And of course, the different nucleic acids are involved in the genetics of biology, the inheritance of various traits of living things, and also the ability to create the proteins that are important for life. But the details of this are very interesting and are what we're going to be discussing today. So let me start by sharing a presentation. This is a presentation on simple biochemistry. Now, I am not a biochemist. I'm not a biologist. I'm not a chemist. But a lot of these ideas are important ones for us to understand regardless of what we study. And in this class, we need to have some understanding of how the atoms and molecules come together to form the biological organisms that we're interested in here on Earth. And that might help us think about what sorts of different biological organisms might exist elsewhere. So we'll start off with simple biochemistry. First of all, we'd like to know something about atoms and molecules. Now, atoms are these objects that were hypothesized to exist for thousands of years, but only really discovered by chemists in the 18th and 19th century as they started to work with the vast array of materials they had available and found that some materials could not be broken down into other materials, while other materials could. And the materials that could not be broken down, they came to understand were made out of one kind of atom. And occasionally you might be interacting with that sort of thing. For example, if you find a 1-cent piece that was made before the year 1982 in the United States, that 1-cent piece will contain mostly copper atoms and not much else. And you can see a cartoon that sort of illustrates that on the upper left. Now, there are other substances which look like they contain only one sort of thing, but you can actually break them down into simpler substances. An example of this is on the bottom left. This is a substance you know as rust. When you take copper and you combine it with oxygen, it turns into a kind of rust, or a kind of patina is another word for it. Now, often people associate the word rust with the oxygen and iron combination. But if you combine oxygen and copper, it will also form a new substance called copper oxide, in this case, copper 2 oxide, that looks very different from the copper you see in the penny, the 1 cent piece above it. And you can see an experiment where the copper is converted into this copper oxide on the right, starting with a bottle full of oxygen in the atmosphere of the bottle and copper on the bottom. Eventually, a reaction will occur and the atoms will bond and combine to form copper 2 oxide on the very far right. Well, how do we know what atoms are and how do we know what makes them up? This was a question that bugged people for many decades, and it wasn't really answered until the very end of the 19th century, when some scientists working together in various locations around the world, but in particular in Great Britain, Canada, and the United States, started working with materials and seeing that there were some ways they could probe the smallest scales of that material. One of the scientists who worked on this was named Ernest Rutherford, and he did a number of different experiments, but one of the experiments he did was to take gold foil, which is some material that only contains one form of atom, and shoot a newly discovered kind of ray at it, a ray of something called alpha particles. Now, when Rutherford was working on this experiment, he didn't know what alpha particles were. We now know that they're the nucleus, the center of helium atoms, but at the time, that wasn't something that was known. What was interesting is that most of the particles that were shot at the gold foil traveled straight through. This was maybe not too surprising because people had found that many sorts of particles and rays that were shot at various objects could travel through them, X-rays being a very famous example. But what was surprising was some of the alpha rays that were being shot at the gold foil seemed to ricochet and come exactly back towards the firing apparatus that was shooting the alpha particles. And this was a little confusing. It didn't take too much calculation for the people learning about this experiment to understand that what this meant was that atoms were made of mostly empty space, but that there was a small and similarly charged, dense object at the center of the atoms, we now call the nucleus, that held most of the mass of the atom, most of the material in the atom. And what happens is every once in a while, the positively charged alpha particle hits that dense nucleus and ricochets back. And that was the explanation for what they saw. On the very bottom on the right, you see what the result is, that we now know that atoms are made of nuclei at the center that are positively charged and surrounded by electrons that are negatively charged. And the scales of nuclei and atoms are wildly different. Nuclei are very small at about 10 to the minus 15 meters across. Well, atoms are also small, but if you look at the scale, it's larger by 5 orders of magnitude, 100,000 times longer than the length across the nucleus. This is much the same as comparing a blueberry on the very far right to a stadium in the middle. This is the same sort of length comparison you might have, imagining the blueberry being the nucleus of an atom and the stadium being the entire atom. That outermost part of the atom is made out of electrons, which have far less mass than the nucleus, but are taking up much more space. However, the electrons are not in the entire space all the time. And so when the alpha particles travel through the gold foil, they're mostly traveling through the empty space of that stadium. Very soon, people began to explore the different sorts of atoms and figure out that the nucleus of these atoms was what determined the different kind of atoms that you had. And there were two particles that were found in the nucleus. One was called the proton, and one was called the neutron. Depending on how many protons you had in the nucleus, that tells you what kind of atom you have, what kind of element you have. And there are many, many elements, more than 100 that are known. And they all have different symbols, some of which are given down below. You can find different lists and explanations of the names for elements and the corresponding symbols. There are lots of ways we talk about the elements and the atoms. For example, we sometimes talk about how many nucleons, that is protons and neutrons, are in the nucleus of an atom. We call that a mass number. And that's a number between 1 all the way up to about 180 or so. And that's a very important measurement, that number of nucleons. We also talk about something called the atomic number, which is a number that runs between 1 and a little bit larger than 100. This is the number of protons that are in an atom. We usually use the letter Z to talk about the atomic number. Now, when physicists think about nuclei and the very center of atoms, they think about the number of neutrons, which they call N, and the number of protons, which they call Z, and they look and they see what those different arrangements of nuclear material do. And on the very left, that's what you see. You see all the different possible arrangements with a black band that extends up from the very bottom to further up along the chart. That black band is indicating the stable nuclei. Those are the nuclei that form the atoms that are around us every day. Unstable nuclei break apart and decay in various ways, don't last very long. And you can see some of them last for a very short amount of time, sometimes less than a second. And there are some nuclei that slowly decay. Those are indicated in the red on this diagram. And in the black, those are the ones, as far as we can tell, that never decay. All of these nuclei are formed in processes in space, processes that were discussed in last week's lecture. If you're curious for where they're coming from, you should go and explore that lecture from last week. As you create these nuclei, you have different amounts of the elements that are created. And at the very bottom right, you can see a diagram that shows the relative abundance, how much of each element there is. But I will warn you that the counting of those elements that you can see on the axis that's vertical on the left is a counting of orders of magnitude. So each level is 10 times more than the level before. This is because the most common elements in the universe are hydrogen and helium. And together they make up 99% of all the atoms. The rest of the atoms are well below 1%. And if you were to try to just count 1, 2, 3, 4, up to 100%, it would be hydrogen and helium, and everything else would look like 0. So instead, we divide by 10 all the way down to the least common element so that you can see what the trend looks like. And that's what we're showing on the bottom right. This is an important diagram for understanding which atoms are more common than others in our universe. When we start to put atoms together, we make things called molecules. And these molecules form chemical compounds that are pure substances that can only be broken apart through chemical reactions, breaking the bonds between atoms. Now, you've been building various models of molecules in our laboratory investigations. And we're showing some here that may seem very similar. At the very top on the left, you see methane. And then you see a version of sulfur, which combines 8 atoms of sulfur together to form a molecule. It can get confusing because sometimes we talk about atoms and sometimes we talk about molecules, and sometimes they have the same name. So for example, we can see in the 3rd row down on the left that you can talk about 1 hydrogen atom or 2 separate hydrogen atoms, or you can combine and bond the 2 hydrogen atoms to form a hydrogen molecule, which is usually designated as H with a tiny little 2 in the subscript next to it. If you had 2 hydrogen molecules, you can see how that is written on the right of that 3rd line. And in fact, there are 4 hydrogen atoms in 2 hydrogen molecules. But then we also have shown on the bottom another substance. This substance is one you may have built in the lab. This is benzene. And benzene is another interesting yet somehow complicated molecule. And you can see what benzene actually looks like in a bottle on the right. On the very far right, we see one more compound that's actually one associated with life. It's called acetic acid. And acetic acid is one of the main ingredients of vinegar. You can see the structural formula and also the model, which includes a double bond to oxygen and then another bond to an oxygen and a hydrogen from one carbon. And then in the other direction, you bond to a carbon, which itself is bonded to 3 other hydrogen. This form we're going to see repeated again. This is called a carboxyl form. It's a form of acid that becomes very important for life. And it's important for life because it forms something called the amino acids, which you can see here. Now, in your laboratory investigation, you were asked to build 1 amino acid, and hopefully that's something that you were able to do. But it's useful to think about what we mean when we say amino acid. An amino acid is a carboxyl group, which is similar to what gives vinegar its acidic flavor on one side, and something called an amino group on the other side. That's a group that uses nitrogen in a form that's similar to ammonia, a simple molecule also important to life. Combining them together with a carbon in the middle produces the general form for an amino acid. Now, what often happens is that other chemicals, other atoms are bonded to that central carbon along with a hydrogen in one direction. And depending on what those other atoms are, you get the different kinds of amino acids that are listed at the left that you can see. You had to build 1, but life builds all of them. If you have 2 different amino acids, as you can see on the top left, they can actually combine together to form a chain. We call that a peptide bond or a peptide chain. What eventually happens is that chain starts to form longer and longer. And each time you form that chain, a little bit of water is released. This is a chemical reaction that allows for bonding between two different amino acids. And the longer you make that chain, the closer you get to what we describe as proteins. On the bottom left, you see a chain that has 7 amino acids together, forming the basis of a protein. Now, sometimes these chains can go on for dozens or even hundreds of amino acids linked together. And then the protein becomes very long and can fold in interesting ways. As the protein folds, it makes shapes that allow for other chemicals to be protected, interact, and sometimes even break within that shape. And that's what you're seeing on the very far bottom right. This is how proteins are behaving in many life forms. Proteins will provoke certain types of chemical reactions. In this case, it's a chemical reaction that breaks down another chemical very efficiently. And we call that sort of action an enzyme action. Enzymes are extremely important things for life and are what allow for some of the processes that wouldn't normally occur in the natural world to occur in living things, and so are often studied by biochemists and molecular biologists. But we're going to think a little bit more generally and not go into too much detail about the different proteins and their functions. Instead, we're going to notice some general features that amino acids and proteins have. And one of those features is something called chirality. This is the same feature that your two hands have. Though they look very similar and have the same parts, you cannot superimpose one hand on another. This is because there's something fundamentally different between your left hand and your right hand. There are other objects which don't have this property. For example, the bottle on the bottom left. If you take a mirror image of that bottle and superimpose it, they match exactly. But hands, your left and right, do not do that. Certain chemicals have that chiral property where they cannot be superimposed on their mirror image, and amino acids are one of them. There you can see on the bottom right an example of a left-handed and a right-handed amino acid. It so happens that life only uses the left-handed version of amino acids. And the reason for that is one that hasn't really been explained. Likely, the first life forms were just using that form, that orientation, that chiral property of amino acids when they formed. But why that is unclear. However, life seems to favor one type of chirality over another. And that might tell us something interesting about what life is and how it works, although it's not clear yet what that fact that we observe is really trying to explain to us. We'll have to do more work exploring this fact to really understand it and come up with a convincing reason why life uses one type of chirality, one type of handedness. Now we'll move on to DNA. DNA being an important other type of molecule for life. Remember, the two types of molecules we're interested for living things are amino acids and nucleic acids. Nucleic acids look much different than amino acids. They're produced by three different parts. There is a part called phosphate, which is the incorporation of phosphorus into living things, very important here. And those phosphate atoms are bonded to oxygen atoms, as you can see. Those phosphates are then bonded to a sugar called ribose. And in DNA, that ribose has an oxygen removed. So instead of a ribose sugar, it's actually a deoxyribose sugar, D meaning removed oxygen from a ribose. So you have phosphorus and you have a deoxyribose, and the type of molecule this is a nucleic acid. So DNA stands for deoxyribose, that's the sugar in it, nucleic acid. And the nucleic acid refers to the different bases, there are 4 of them listed here, that can attach to that sugar. And the different bases are made out of nitrogen and carbon and hydrogen and oxygen, and you can see what they look like here. They have different names: adenine, thymine, guanine, and cytosine. But because scientists are lazy, they often abbreviate those bases with just the letters A, T, G, C. So you'll often see the code for DNA written out one letter at a time, telling you what the order is for these bases. And there are trillions of bases in every genetic code for every life form. So while the chemicals are bonding in the way you see on the left, because it becomes very complicated, most people are familiar with the image on the top right. That's the image of the double helix. And this is exactly the way this phosphate backbone connected to a deoxyribose sugar connected to a base will fold and turn as it is coiled up and stored inside a cell. What you should understand, though, is that shape is standing in for those atoms bonded together. The phosphates on the outside connected to some sugars, and the bases on the inside. The bases don't actually bond with each other in the same way that you were modeling the bonding of atoms when you were investigating molecular models. They bond through something called hydrogen bonding, which is a very weak kind of bond that's easily broken. And that's actually an important feature of DNA, that easy to break the rungs of the ladder inside of it is an important thing to be able to do, so that other molecules can get the information that's stored by the order of those bases that are in the DNA molecule. Now, you might be wondering how we figured all of this out. Atoms are incredibly small, and we've only in the last few decades figured out how to image them directly using things like scanning tunneling microscopes. Well, it was a clever game that was played. About 70 years ago, people were using X-rays to examine lots of different things, including crystals. And when they noticed that different crystals would reflect and refract, that is, bend X-rays in different ways, they could figure out by the way the X-rays were bent what the arrangements of atoms were in the crystal. This was a pretty remarkable feat, and it was an entire study that people would do called X-ray crystallography, which means the drawing of or writing down of crystals using X-rays, explaining and being able to calculate the different arrangements of atoms and crystals using X-rays. Well, people knew that nucleic acids, and in particular this nucleic acid, DNA, seemed to be a very important molecule for living things. But they didn't know what shape DNA had, and they had a hard time figuring it out because it was so gigantic and had so many base pairs. It was hard to know what was going on. But there was work that was done by a woman named Rosalind Franklin, who's shown on the very far right, where she passed the solidified DNA with some X-rays that went through it and took the very famous picture you see next to her. And this picture can be interpreted to explain the shape of DNA. I won't go into the details of how that works, but the other people that were working with her, James Watson and Francis Crick, were able to figure out that this indicated that DNA was actually a double helix coiled on itself. And for that, they won the Nobel Prize. Rosalind Franklin should have won it as well, but she wasn't granted it by the Nobel Prize Committee. So how does this work? Well, in your cells, you have a small little part of it called the ribosome. And the ribosome is able to separate out one strand of the double helix of DNA and feed it through this little unit. There's another form of nucleic acid called messenger RNA, mRNA, which actually takes the information from the DNA and then passes it through the ribosome. As that passes through the ribosome, other forms of nucleic acid RNA, we call it tRNA or transfer RNA, are able to enter into the ribosome and then find particular patterns in that messenger RNA that indicate a kind of amino acid that should attach to the other end. So the RNA can attach to both nucleic acids and it can attach to amino acids. And as it does this, it can allow for those amino acids to link together. And this is what forms proteins in your cells. So the proteins that are produced by living things, at least those that have ribosomes, all go through this process. Now, there are other living things that don't have ribosomes, but they have the same sort of structure where the RNA takes information sometimes from DNA, but sometimes it's just stray RNA that comes through. And as it passes through these areas where the tRNA can attach to it, that's where different forms of amino acids will start to bond together to form these long, we call them polypeptide chains. And when that occurs, you start to create the important proteins for life. Now, in your body, you have RNA and you have DNA, but there are occasions where foreign DNA and RNA can enter in and disrupt the process. This is, for example, how many viruses work. And so that's an interesting thing to keep in mind, that these processes, they work, but they're not things that are foolproof. This is just something that follows the pattern due to the bonding and the attractions and the movements of atoms and molecules within these cells. So that's where we will leave our understanding of biochemistry. What is happening is we have a recipe card that's kept in the DNA that says what types of proteins will be created. And then we have RNA acting as kind of the bridge between the amino acids and the DNA. And finally, we have these long chains of amino acids produced, which form proteins, which do all of the chemical reactions and help with the enzyme actions that we usually associate with living things. That's where digestion happens. It's where photosynthesis, the creation of energy by plants, happens. This is what makes life life. So this is why we rely on this biochemical definition. It seems to be very important for all living things that we see. As we move forward, we'll see that even though living things share this very unique and actually pretty simple, when it comes down to it, you're only using 6 types of atoms to do all of this work, process, even though this process is shared, there's a lot of difference and diversity among all the living things. And that is something we will explore and explain quite a bit more as we move forward. So that is something to look for and to look forward to for next week. Keep in mind, as you start to read and learn about these things, that there is one molecule that we mentioned here that forms a bridge between proteins and nucleic acids, and it is a nucleic acid itself. It's called RNA. RNA stands for ribonucleic acid. So deoxyribonucleic acid uses a sugar that removes the oxygen. If you don't remove that oxygen, you form this other nucleic acid called ribonucleic acid, which uses ribose with the oxygen still there, but pretty much the same as DNA otherwise. And this molecule may be the key to understanding how life first emerged. And there are some interesting ideas about RNA being the original flavor chemical for living things. We'll explore that more next week. So that is as far as we have for this week, our discussion. The reading that you can do is the same reading as from last week. So a lot of the material that we've been discussing over the course of these 2 weeks are kept in those 90 pages worth of material. So I encourage you to read through those, to try to understand it, and hopefully that will help you answer some of the questions that you might have when we are talking about what the biochemical basis for life is. Have a wonderful day. I hope you enjoy learning about these things, and we'll see you in 1 more week.